if you are on the European time, and welcome to the second of the education webinars provided by the European Society of Mycobacteriology and supported by Becton and Dickinson. Today, our speakers are two excellent scientists, Leen from the Institute of Tropical Medicine in Antwerp, and Stefan Niemann from the National Reference Center for Mycobacteria in Borstel. The topic of today's seminar is tuberculosis drug susceptibility testing, genotyping versus phenotypic approaches. What and how, advantages and limitation. So I think it's going to be a very hot topic for all of us. Let me first introduce the speakers and then move to few housekeeping tips. Stefan Niemann is the head of the molecular experimental mycobacteriology in Borstel, Germany. The topic of his research are Mycobacterium tuberculosis complex strains and other bacterial lung pathogens, is focusing on local and global transmission dynamics, determination of resistance and compensatory mutation mechanism, population structure, and evolution of MTB strains and other mycobacteria. It's also focusing on virulence, physiology, pathobiology of mycobacteria, and application of high throughput technologies in research and diagnostics, implementation of new technologies in high incidence countries, as well as for personalized therapy and evolutionary medicine. Leen joined instead the mycobacteriology unit of the Institute of Tropical Medicine in Antwerp. She is a worldwide known expert in detection, isolation, identification, and drug susceptibility testing of mycobacteria using both classical microbiological and molecular biology tests according to good laboratory practices. Since 2011, she manages the BCCM-ITM collection for mycobacterial strains. She's also expert working with the WHO in many high-burden tuberculosis countries. So we do have two excellent speakers for today. Now let me move to the next slide, and with very few housekeeping tips. So you can send us your question during the talk using the live chat window. To use the live chat, you'll need to sign in YouTube with either YouTube or Google account and join the channel. Include your affiliation in your question. This is because if we don't have time to answer, we will get back to you. And the chat, I will personally look at the chat and then refer the questions to the speakers. I will remind you that these seminars are kindly offered in collaboration with Becton Dickinson, and with that, I'll ask Leen to start her presentation. Leen, the floor is yours.
Good afternoon, everybody. So my talk will be about phenotypic drug susceptibility testing, and I try to answer the question whether this is still relevant now with the outcome of the molecular testing. So basically, you can test either the minimum inhibitory concentration of the bacilli, which is defined as the lowest concentration of an antimicrobial that will inhibit the visible growth of an organism. And typically, we test at least 5 concentrations with 2-fold dilutions. In bacteriology, often they test whether at which concentration 50% or 90% of the bacilli are killed, which is then referred to as MIC 50 or 90. But in TB, we typically test MIC 99. And this is mostly associated with the testing of the standard phenotypic testing, and which is usually done by the proportion method. And this method allows growth of less than 1% of the bacilli at a given critical concentration. And usually there is only one concentration tested, like for rifampicin, this is 40 microgram per milliliter on Leuwenstein-Jensen medium. And I'm sure you're all acquainted with this methodology in which you inoculate your slope and an antibiotic-containing slope and a control slope with a 1 in 100 dilution. So you can see if there is less or more growth to define whether your strain is susceptible or resistant. This takes up to 4 to 6 weeks of growth. And so this is a quite slow method, which is in place for decades. And in 2005, WHO endorsed the midget system as a rapid broth-based medium looking at the metabolic activity. And you define susceptible and resistant if you have more or less than 100 growth units of growth. In 2011, the WHO has endorsed more rapid phenotypic DST methods, amongst others the nitrate reductase assay, the REMA method, and the MOTS. The MOTS is a microscopy-based method. From all of these, the MOTS can be done directly on clinical specimens, so without the need of primary isolation of the bacilli. There are still some more rapid alternative methods, but they are not endorsed by WHO, mostly because there is insufficient data to do so. And from these, the thin layer agar can also be done directly from the clinical specimen, saving time. The sensititer plate is now being more and more used, and there recently have been some multi-center studies applying this method. I will come back to that. Phage-based typing is not that much used. These rapid methods have the advantage that it can be high throughput, and they are more suitable to do MIC testing because you need, of course, less space in your incubator and so on. But they are rarely implemented in routine and programmatic settings. So there is little data available from these tests. So they are more at this time point research applications than used for patient management, which also gives that there's less certainty around the critical concentrations for many drugs in these assays. Now, the internationally referred standard method, reference method by EUCAT and ESMIC is in Middlebrook 7H9 broth on a microtiter assay by visual inspection. So no color indicator is used in this assay typically. Again, this is not used in many laboratories and only few have experience with it. So how are the critical concentrations and the breakpoints defined? In the early 1960s, by Canetti and colleagues, there were some important publications on this, and they defined the critical concentrations based on clinical observations. But the WHO has regularly updated these recommendations, and the latest updates are from 2012 and 2018. In 2018, this was based on a systematic review of literature data. And based on that, some recommendations were made, but not for all drugs. They were not all revised. So there are still some gaps. If you look at the EUCAT recommendations, there is limited guidance for tuberculosis, Mycobacterium tuberculosis. At the 2020 recommendation, you can see that there's only a critical concentration defined for delamanid and bedaquiline. In bacteriology, typically, the system of susceptible, intermediate, and resistant is used, while in tuberculosis, usually we only use resistant or susceptible. And a strain is defined as susceptible if there's a high likelihood of therapeutic success using that concentration. Whereas it is considered resistant if there's a high likelihood of therapeutic failure. The intermediates are the susceptible at an increased exposure, which is now the new name given, can make a strain again susceptible if a higher dose of the drug is given and can be given in a safe way. Now, to define whether a drug at a given concentration is effective or not is very complex in TB treatment because we always give a combination therapy. So the role of each drug in the treatment success is difficult to define. So what is done mostly in the tuberculosis is we look at the Ekov, the epidemiological cutoff or the microbiological cutoff. So if you look at the wild type population and the distribution of the MICs against those of the non-wild type, the mutant population, you normally have a bimodal distribution. And you can differentiate the wild type from the non-wild type population. And the Ekov is defined as the MIC value of the upper limit of the wild type population of naive non-exposed strains. This data to define the Ekov for a specific organism, for a specific drug, should come from at least 100 strains tested and preferably from different laboratories. Also, all different lineages within the MTB complex should be used in defining the Ekov. The Ekov is not always that clear. Sometimes there is some overlap, like this is the case for ethambutol. Also, within those that have the non-wild type population, you can have different mechanisms of resistance. And I'm using here a slide from Claudio Koeser, which nicely explains the concept of susceptible and resistance based on this distribution. So if you find in case A and B here, you have the susceptible strains. They are below the Ekov, which is the wild type population of your organism. And this is the case, for instance, for isoniazid in non-tuberculous mycobacteria. This is a non-effective drug for many of the non-tuberculous mycobacteria. Sorry. It's also possible that you can define an Ekov, and the Ekov coincides with the susceptible and the resistant population, the non-wild type population, which is the case for most anti-TB drugs. And that's also why we usually test a single concentration, the critical concentration. Now, in some cases, you have a bimodal curve following the Ekov. So you have mechanism A and mechanism B, and which could allow you to define a critical concentration and a clinical breakpoint, or to define it as the susceptible, the intermediate, or the susceptible at an increased dose, and the resistant ones. This has been shown for the fluoroquinolones and potentially is also true for isoniazid. What is specific for the Ekov? The Ekov does not change by the sampling. If you would test samples from Brazil or from North America or from China, they should all show a similar Ekov. And what is true is that the Ekov may change by testing medium. We all know that the concentrations in MJIT on LJ or an agar medium can be different. So also the Ekov may differ. The clinical breakpoints, however, may change if there is new resistance mechanisms occurring in the bacilli, or if there is an altering dosing used and different indications for the drug use. If we say that fluoroquinolones can be given at a higher dose, and we do have a bimodal curve, we can obtain a new clinical breakpoint for the organism. And we have shown it's Armand van Deun in a study in Bangladesh, has shown in MDR-TB treatment regimen, the short MDR-TB treatment regimen, that patients with susceptible fluoroquinolones had a good treatment outcome. Those with low-level fluoroquinolone resistance also still had a good clinical outcome with no significant difference, while those with high-level fluoroquinolone resistance had a worse, much worse outcome. So there seems to be a difference between low-level and high-level resistance in the treatment outcome, which is also linked to the molecular mechanism behind. And probably Stefan Niemann will tell you more about this. So this is why we could define for the fluoroquinolones, the susceptible strains, those with an intermediate result in treatment outcome, and those with a clearly resistant profile, the high-level resistant ones. And this here, the success depends on which dose of treatment is, dose of the drugs given. But of course, it's not only the MIC distribution that is relevant to define whether there's success of treatment success or not. And also, we cannot just increase the dose of each single drug. Therefore, the PK/PD data are equally relevant. So the pharmacokinetics defines the movement of the drug through the body. So it's in function of the time, and it mainly describes the bioavailability of the drug in the body. Does the drug get where it is needed in the tissues and for a long enough time? While the pharmacodynamics is more related to the body's biological response to the drugs. Can the drug do what it needs to do? It is inactivating or killing the bacilli without doing too much damage. So both the PK and PD data should be taken in account as well. And it is based on the difference observations of the bimodal curve for fluoroquinolones of susceptible ones, low-level resistant ones, and high-level resistance ones, linked to the difference in treatment outcome and linked to the PK/PD data that for the Moxifloxacin, for instance, it was decided to define a critical concentration and a clinical breakpoint. This is actually the only drug for which this is done at this moment. As I told you already, the Ekov and the critical concentration depend on the medium used. In this study here from Isaiah, you can see that the MIC for ofloxacin decreased when midget was used compared to Löwenstein. So it is very important in publications and in reports to always mention the medium used when you show MIC data. Also, for rifampicin, for instance, which is very strongly protein binding, the critical concentration in Löwenstein is much higher than the ones in agar and broth medium. We also have the phenomenon of phenotypic cross-resistance. So we could ask ourselves, if we have a class of drugs, can we test just a single drug from this class to have a representative idea about the resistance level? And this is a publication from Farah Marat from 2015. And this clearly shows that there is cross-resistance between moxifloxacin and ofloxacin. And they only differ in the level of MIC, but there is cross-resistance. And they have shown that testing ofloxacin as a surrogate for moxifloxacin resistance gave a high sensitivity and specificity. So there is no need to test each single fluoroquinolone in your lab. You may also test just one of the class. But phenotypic DST, although it is still considered the gold standard, needs further standardization. And the things I've written here in gray, I think you are all familiar with the need for EQA and internal quality control. What I wanted to highlight mostly is when you do MIC testing, we all know that you can accept one dilution variability between two results to consider it as still being the same result. But we also have the gray zones, especially in the rapid methods and the microtiter plates, for instance. You have these precipitation that is not always that clear. You have the blue and pink in REMA versus the purple. So there is some sometimes difficulty in making the interpretation. And this is a recent publication from Ismail and colleagues on the use of this microtiter plate testing on large scale using prepared plates. They used dry and frozen plates, and they didn't see a significant difference between the performance of both plates. but there was clearly some interlaboratory variability, and it is a complex figure that is shown here, but you may take a look at it yourself later on. But it's clear that the different laboratories had different peaks, and there was some variability between the laboratories. Phenotypic TST is the gold standard, but it's also prone to errors. And this is well known for PZA testing, for instance. In a recent publication from Claudio Köser and colleagues, where they looked at the MIC testing and the mutations in gyrase A gene, they proposed to testing, in addition to the classical 100 microgram per milliliter, also to test 50 microgram per milliliter and come to the classification of susceptible, resistant, and uncertain in case you have an MIC of 100. And there you may have additional PNCA sequencing that could make a final clue on whether you have a PZA resistant or susceptible strain. And this is mostly because you have almost overlapping, you have slight overlap of wild type and mutant populations. Also, the procedures that you follow should be followed very strictly. And Sven Hoffner and colleagues have shown that in using midget PZA, only after training and highlighting, pointing out some important things like the good inoculation volume, they obtained better results in Sweden. But also the rapid tests may miss some mutants, and this is well known for the rifampicin so-called elusive mutations. And a recent publication from Gabriela Torreya and colleagues, you can see that the proportion method here, when you look at the wild type strains, all were correct. When you look at the most common high confident mutation of the 450 leucine, all were detected by the proportion method and by most of the rapid methods. But if you take the elusive mutants, then they are having a lower level of rifampicin resistance, and they are very often missed in these rapid methods. So you could also question, what about the EUCAT standard microtiter plate assay? Maybe we have to revise again using these elusive mutations. So heteroresistance is also an issue in Mycobacterium tuberculosis, either from having two different infections or from an evolving resistance, and also the different sputa that can have some of these susceptible and some of the resistant strains. But the limit of detection for the heteroresistance is very good for phenotypic DST and better than most of the rapid molecular testing, at least. So in principle, we can detect up to 1% of the variant resistant mutants. So there is still some issues for many of the less common drugs from group B and C. There is no recommendations, mostly because there's insufficient evidence and no MIC data to support it. On the other hand, we have the new and repurposed drugs like bedaquiline, clofazimine, and delamanid, and there are some recommendations that are made on the limited MIC data available. So there is tentative critical concentrations that have been proposed, and they are mostly defined based on the Ekov. But what about pretonamid, for instance? So there's still some work to be done. And why my showing here is already something from the genomics, which Stefan Niemann will talk about. But for the new drugs especially, the association between phenotypic and genotypic testing remains puzzling. And this is a recent publication that has been accepted last week from Battaglia and colleagues. And this is showing here all possible variants in genes associated with bedaquiline resistance, like the RV0678 gene, and with the different colors represent different lineages. And this is from a study from 5 countries where a drug resistance survey was done. So these isolates basically were never exposed to bedaquiline or delamanid, and probably not to clofazimine either. And we can still observe a wide variety of these mutants. But what's also striking, and maybe it's not that clear here, but only the red triangles had elevated MIC levels, and the yellow ones had intermediate increase of MIC levels. All the green ones didn't show increased MIC levels. So it is puzzling, yeah? So that's why phenotypic testing, especially for these new drugs, is still needed. So phenotypic DST is still relevant, but we still need more standardization. So the EUCAT microtiter plate should be used as a reference, but we also have to think about these potentially missed resistance for elusive mutations. But we definitely need more Ekov data. And what is also needed is more MIC data that can be linked to molecular data, to treatment outcome data, and to PK/PD data, because that's how critical concentrations and potentially clinical breakpoints should be defined. And we need this for the new drugs, for the repurposed drugs, but also still for some of the old drugs where we are still puzzling about the significance of molecular data. And I think we've still got work to do, and I thank you for your attention. And I'm happy to give you the floor now to Stefan Niemann.
Thank you, Leen. And now is Stefan. If you can share your screen and go ahead with your presentation. Stefan, the floor is yours.
Thank you very much. It's a pleasure for me to present you some thoughts about genome sequencing and how we can use it for resistance diagnostics. Actually, the concept was born quite a while ago, and this is just from a review that actually suggests that with the advance of genome sequencing technologies, the genome can be sequenced for particular pathogens in routine diagnostic workflows, and you can basically address quite a number of questions by the sequencing that comes with the species identification over relatedness, which is not the topic for today. That means cluster analysis and transmission. And then you can look into resistance and into virulence. Knowledge databases to judge or classify the isolates. And that's, of course, would be one technology used for all these technologies. And that's like a paradigm change in clinical microbiology by one breakthrough technology. So in TB, particularly, we would, of course, like to do better diagnostics. We want to move to personalized treatment regimens early on and link that to high precision outbreak analysis in genome-based molecular surveillance. And that was already the topic last week. So one particular problem is that, for example, for the new medicines that are used for treatment of MDR TB, currently there are a number of drugs, or quite a substantial number of drugs, so to say, where either no molecular or no robust phenotypic test assay is available when you consider it on a worldwide level, and therefore universal access to drug susceptibility testing is far from reality, and the majority of patients for which these drugs are used are treated blindly. And I think is in a way unacceptable and also has a high risk of treatment failure in those patients that may be resistant. So here other approaches like rapid molecular tests are basically essential to solve that. That is the concept that we propose, is actually that from the genome sequencing data, you basically map normally the reads generated to a reference genome sequence. By that, we get variants, and the variants are going into an interpretation pipeline, and then you have like high-level resistance SNPs or variants, and in those situations you cannot use the drug anymore. You may have then the situation that you know that some variants just induce a lower level of resistance, and in this case you can adjust the dose potentially or you may have variants in resistant genes, and that's a substantial number of genetic changes in those genes that are phylogenetic, so that are not causing resistance. So in this case, of course, you can still use the drug, and in a way, this information is going to do individualized treatment of at least MDR and pre-XDR and XDR TB cases based on the genomics of the pathogen. And in addition, we can use this data for integrated molecular surveillance systems. So if we then look into the regions involved in resistance, then we do see, even on this slide here, that there is a high number of genes already when we would make a short list. But actually, in our data analysis, we are currently looking into more than 3,000 variants in more than 90 regions that are potentially involved in resistance. And though the variants that we qualify are resistance-associated variants, but we also have mutations in those genes that are benign, so not leading to resistance. But if we want to use the data to predict resistance and susceptibility, we would need to have a knowledge base that can do both, or classify both mutations in either resistance-associated or not resistance-associated. So molecular resistance prediction per se is not a new technology. So we started already in 2001 and early on to use classical Sanger sequencing. And already there we started to understand resistance mechanism. In this case is rifampicin, but also of other drugs, although this was basically more cumbersome as using genome sequencing. In 2005, we basically did our first paper on using Hein line probe assays that were developed at that time to basically allow like a reverse hybridization assay and detect mutations related to resistance. But that is of course quite restricted to certain areas of the genome. And in 2009, we then basically did our first paper, or the first paper at all, that looked into or used Illumina sequencing for describing the genome of just 2 TB strains at that time. And further on, we of course developed our concepts for using massive genome sequencing in clinical diagnostics, and started testing the correlation between genotype and phenotypic resistance predictions to learn how these tools can be used. So what is genome sequencing? And I'm not going through this cartoon in detail, but there are basically three major sequencing technologies, so to say, or concepts. The first was Sanger sequencing. That is what we have used around 2005 and before, where you have basically amplified a particular gene, and then you can sequence this gene, you get the sequence information, and you can use this information to define SNPs, and then you have the same information basically then for the second generation, then a next generation sequencing technology, with the difference that next generation sequencing is actually looking into the full genome, and it's gonna somehow fragment the genome. It's put on a surface, amplified, and then it's basically sequenced with a comparable technology than the Sanger sequencing, so it's fluorescence based, and you get millions of short reads that then can be further processed. And we now have long read sequencing that is a bit different, because it's not going to deliver short sequences, it's delivering long reads that has certain advantages. And this is one machine that we just got in the laboratories. It's a PacBio Sequel 2 system that can produce long reads, but there is also a small machine that is a MinION, and this is quite promising for field work, because as you can see here, you can even sequence in a driving car with this technology. The basis, there are now benchtop systems available, as you can see here, for normal lab integration, and especially the iSeq system on the right is a highly versatile and small system that is also not too expensive. And the MinION system is not Illumina, it's a different technology, but also can be used in the field. The major differences here that you need to consider when you start planning your sequencing laboratory are actually shown here. And there are two basic things. One is the read length. So the read length of the sequence data you get out has implications for the data analysis stream, but also for the questions you can address with those data sets. So very long reads can be generated with a MinION and with a SQL system. But if you consider output, for example, then the different Illumina machines also deliver different outputs, and the outputs, that is basically describing the number of strains or the number of samples that can be analyzed in parallel. For example, on the NextSeq, we sequence roughly around 200 TB strains per run. On the MySeq, it's then 30, and on the MiniSeq, it's even lower. So that has also implications for the cost per sample, but also for the output that you need to run such a machine efficiently. So for the NextSeq, you at least have to put like 100 samples on the machine, while on the MiniSeq, this is much lower. And for clinical diagnostics, this of course has some implications. If you are in a high incidence area with lots of samples, you need a larger machine, while if you just have few samples, then you can go for a smaller one. Just one word to the long read sequences. We use this mainly for looking into other bacteria. So for example, in non-tuberculous mycobacteria and other resistant bacteria, to also reconstruct for example, plasmids or to reconstruct the full genomes. Therefore, the longer reads are quite essential. So major differences, cost per sample, read length, and error profile, and that of course needs to be considered for your planning, but also for your data analysis. So this cartoon should just show you that, I mean, at the end, the lab workflow for classical Illumina sequencing, this is what is shown here, what we set up in our laboratory, is basically not as simple as one may think, but I must also say it can be established also in different flavors, and it is, we also managed to establish the workflow for whole genome sequencing in high incidence settings. And the charm with Illumina is that you can start from low input DNAs that, for example, are coming from lysates. So when you don't have a really cleaned up DNA, there is other possibilities, and you can basically go with the same pipeline in different sequencing technologies. So like the MiniSeq, the MySeq, and the NextSeq, and also the iSeq, and it's all going through more or less the same analytical pipeline. And what we also have developed is an automatically created next generation sequencing resistance report that you can see here. That is coming directly out of our analytical pipeline and can then manually be expected. So saying that, this is also just a slide that should show you that if you do NextSeq runs with 200 samples, the whole library preparation workflow also requires quite a substantial amount of pipetting. And I mean, this is actually just an illustration of the work that is still related to sequencing when you do larger sample amounts. The charm with the overall technology is that you can do TB, but you also can do quite a number of different bacteria. That is interesting when you set up a core facility, for example, that can then also be used for like Maltophilia or Abscessus, Kansasi, Chimera, and other bacteria. So the data analysis tools, this is, I think, one of the most important parts in next generation sequencing applications. And there are now tools out that can be are basically pre-defined and can be downloaded and installed and run more or less automatically. One is our pipeline MTBSec that basically is doing all the steps we have defined as high or as needed for guaranteeing a high quality output for your data analysis in a row. So that is, I think, working quite nicely. We also have a low frequency mode for resistance prediction, because normally for outbreak analysis we would not really consider low frequency calls, but in this case for resistance predictions we need that, and this is available via GitHub, and I think a number of labs around the world are currently using that already. There is another version for just resistance prediction data analysis, and this is a tool that we developed quite some time ago. That's called Virus. This is available in the lab and that is in the web. This is fully automated, so you can just upload your file and all the steps are done in the background, and you just get a notification when the results are ready and you get a resistance report. We are currently working on a new version and to implement our new resistance mutation catalog, and we're also working on a version that can take amplicon data. So what is now our knowledge on resistance mutations and on mutations that may not confer resistance? This is an example, or is one of the first attempts to get a curated catalog, an accepted catalog of resistance mutations. This was published by, or was done in a large collaborative framework, but done by our friend Paolo Miotto. And basically this was a starting point where we proposed a first validated list of 286 resistance mutations. And I guess that from there, then the next landmark study was the study of the Cryptic Consortium, where we looked in 10,000 genomes and that were collected among 16 countries across the continent. And the most important statement here, that the data that we got supported the conclusion that whole genome sequencing can now characterize profiles of susceptibility and resistance to first-line drugs with a degree of accuracy sufficient for clinical use. And that was also the starting point of the Public Health England to basically switch completely towards resistance, towards whole genome sequencing based resistance testing. And also a number of other labs are now completely changing their procedures, at least for first-line drugs, and for second-line drugs, the relevant studies are on the way. There is one important notion that we should know, and this is a study that we recently published, where we looked into the diversity in 92 genes that are potentially associated to 21 drugs, and we used a reference collection of 405 strains. And what you can see on the right-hand side is a phylogeny based on the sequence variation only in those genes. And the striking result was that you could find enough variation in the resistant genes to completely rebuild the phylogeny of the TB complex. And we overall, we were able to find 435 phylogenetic mutations. We were not yet able to classify all of them as benign, so not confirming resistance, but it shows that these studies are essential to really interpret whole genome sequencing data, as we do find significant phylogenetic variation in resistant genes. Some of this indeed could lead to resistance. So there is a proteonomide resistance in a lineage 4 clade, and there may also be clades in the TB complex that are hypersusceptible to beta-caline due to particular mutations. But what is clear is that this needs to be included in our interpretation catalogs. So if we look into sequencing workflows and what we can do currently, then it is, in my view, established that from sputum, we can mainly do targeted sequencing, and I will explain a bit later what that means. And the majority of studies or approaches that use whole genome sequencing, so the extraction of full genome, are done from culture. And overall, that all should lead into a plain language report. So if we work from sputum, there are two different technologies or different technologies. I would shortly go into specific enrichment. And the most easy enrichment is a short culture. And a short culture is still an option. And in the study we actually published quite some years ago, we worked from early positive liquid cultures. And the first result that at that time was not, as predicted or predictable as it would be now, where we could actually make quite a number of whole genome sequencing resistance predictions that were to a high degree concordant with classical DST data. And we made additional whole genome sequencing predictions for second-line drugs that were not discovered by the classical phenotypic assays because they were not performed for non-MDR strains. So overall, the whole genome sequencing reports could be generated in a median of 9 days, and that was much faster than the classical reports were done. So you can actually see, even if you have a culture step in, you can still be faster than classical technologies. What I wanted to show you here, that even when you use an early midget culture, you have quite a substantial amount of other genetic material in these data, and this is shown here. This is mapped to M. tuberculosis, and this is mapped to other stuff like human or non-pharyngeal flora. And this diversity in the sample can actually interfere with your results, and you need to account for that when you work on those samples. Similarly, this is direct sputum sequencing. When you deal with direct sputum sequencing, first of all, you have a high number of samples that contain human reads and other reads. And overall, current approaches are actually allowing a prediction of resistance in just a fraction of those samples. There is a new paper from the same group, but it basically has the same problem, that you can do it, but the coverage is quite low for quite a number of samples. So here we need to get much better. There is another approach that is specific amplification of resistant genes, and then you would just look into that. And there's actually one assay that is out, that is a duplex assay that basically comes from the genome. Then you do extraction, amplification, data sequencing, and data analysis in a cloud-based system, and you can then go to resistance report and individualized treatment. That looks into quite a number of resistant genes, as shown in this cartoon. And we just did a first evaluation with this setup here. So from the sputum, we basically did a decontamination, cleanup, and then a workflow for the library prep and the sequencing. So that is the automated data analysis in the cloud that comes with a quite nice layout and interpretation catalog. And when we did it, we looked into 50 clinical samples, and 39 of those yielded interpretable duplex result. And the main finding here is that basically the successful, so everything that was smear positive could be more or less or to a high degree produced an interpretable result. Interestingly, is that this was not only true for sputum, this was also true for other tissues. So we may have here a tool that can be used directly from sputum in quite short time. So the midget-based whole genome sequencing workflow could potentially lead to a result within a 10 days workflow. But if we do the targeted sequencing from sputum, we get to an interpretable resistance result within 5 days. And I think this is quite a speed up of the time to diagnostic at a high accuracy. So in summary, I have shown you that whole genome sequencing has a high performance for predicting susceptibility and resistance. We can do it from early positive midget cultures, and targeted targeted approaches from sputum are really a valid option. There is something to do. So the limitations are that I think the NGS from sputum needs to be optimized. Mutations catalogs still need to be further improved. And the whole genome sequencing workflows and data analysis workflows are complicated. And the infrastructure and personal competence needs have been developed at sites. And we also need to get better in DNA extraction, enrichment methods, and workflows for high incidence settings. With that, I just want to thank my group and all collaboration partners and you for your attention.
Thank you very much, Stefan. That was excellent, as well as was excellent Leen presentation. And now the floor is open for the questions. We do have the first question for Leen. And the question is from Olena from the UMEA University. And the question, Leen, is what are the main reasons for testing variation between laboratories?
Oh, I think that's a difficult question. There may be slight variations in the different compounds that you use, the drug compound that you use from a different brand, the medium that you use, how you prepare it, slight variations if you have homemade medium. And then, of course, it is, we are working with viable bacilli, we're working with tuberculosis, which is clumping. So the preparation of the inoculum is very important. And the way that you make your suspension and the subsequent dilutions are really crucial. And that needs really hands-on training and participating in EQA, and you have good results probably indicates that you are doing it well, but otherwise you may have to train each staff member again and again and making sure that everybody is participating in EQA on a regular time. So I think it's partly the materials that can slightly differ, but also the manipulation of the bacterial suspension, which is really crucial.
Thank you, Leen. That was a very clear answer. And we are waiting for more questions. We still have 8 minutes to go. And while we are waiting, I would like to encourage everybody. We have more than 100 people connected. So please send your question and the speakers will give you the answers. And I do have a clarification for everybody. You mentioned the EUCAT protocol, but saying that this is maybe difficult. Few laboratories are able to do that. I just want to make clear for everybody that this is going to be the standard protocol to establish the Ekov for new drugs. And so this is not a protocol that will need to be implemented in routine laboratories. This is going to be the standard to use as a comparison for all the other methods that are commercially available. So what is going to be the big work that will start very soon is the standardization of the current method against the standard. So once this is done, then the commercial method will be equivalent to the standard one. So it is not going to be a method that we will ask anyone to implement. It will only be the method that will be requested by the EMA for the new compounds to really make sure that what are the tentative Ekov are established with a standard. This is just a clarification.
Thank you for that, Daniela. I fully agree.
Stefan, I do have a question for you. And this is whole genome sequencing or next generation sequencing technologies are really appealing, but more and more people keep arguing, saying that the application in high burden setting is going to be too challenging to become a reality. And I know that you have comments that you can share with us.
Okay, that is a complex point, and in my view, it is possible to implement that in high incidence settings. We did it in Kyrgyzstan, where a MySeq is running, and we are currently implementing sequencing in Namibia and in Eswatini, but there are challenges with the supply chains, for example. It's not essentially the people on the ground. That is another challenge, of course. You have to have trainers that go there, people that are willing to stay longer than 2 days in countries to support on site. And we just are about to learn how we can get everything in a kind of package in the countries to have longitudinal support from the sequencing companies to convince them to do that. But the next step, I think, that is essential is that the sequencing reagents get into the reimbursement catalogs, I think, of the Global Fund, for example, so that the companies can also have access to the chemicals, similar to that they have access to midget, for example. If that step is done, then it also gets much easier to integrate these strategies into their local TB control plans and into the local algorithms.
Thank you, Stefan. You do have other two questions that are coming now. One is from Maxime Godefroy. It says, Stefan, I assume all analyses are done with H37RV as a reference. Will there be a movement forward towards using another reference or a more precise reference according to the strains analyzed?
I mean, so for resistance prediction, we use the resistance gene or the genes from the H37RV genome currently. Of course, if there would be an example that there is an additional resistance gene that is not in H37RV, so you would find a resistance gene in Beijing strains, for example, and it's not existing in H37RV, we have to include it in our analysis pipelines. Currently, the standardized way is to look into H37RV, and all mutation catalogs with the positions of the mutations are based on that. And it's strongly recommended not to change it, at least not for resistance predictions.
Now you have a question from Moscow. And this is, I think that will open up a little bit of a discussion. And is Danila Zimenkov that is asking, we find in an RPOB L511R substitution minimal confidence according to Paolo 2017. Can we still prescribe rifampicin?
So that is a question that
I go and Leen goes.
Yeah, so that is a question we should discuss offline. She can just send me a mail and we're going to look into our advanced interpretation catalog. And likewise, we would then also take Daniela in the round to answer that question. In particular, I must say that for us, in the majority of MDR strains or the MDR cases that we currently prospectively investigating with whole genome sequence in our clinic, the majority of mutations are clear-cut. But the examples, or for some mutations that are at low frequency, we do not have enough statistical power to look at that. And there are some mutations, like, for example, L430P, that are constantly tested susceptible with the midget, but are actually among the mutations Leen just described that are testing resistant with the Löwenstein-Jensen, and there are also problems with interpretation of those. So here we would overrule midget and would say this is a resistant mutation and don't use the drug. If you could potentially use a higher dose of the drug is something that actually also needs to be proven at best in clinical trials. Those one we don't have currently.
Yeah, I mean, if I can add that as a general comment, the minimal confidence mutations were minimal confidence because they had a very little representation on the global catalog. The fact that they were associated with a confidence grading were because they were testing resistant in the majority of cases. The other point is that currently, the midget testing is a little bit overestimated, so WHO may reconsider that. Right now, as a general rule, mutation that have some confidence grading need to be considered resistance, but then we will discuss definitely this offline with Paolo as well,
yes.
that is collecting more data that is next door. And should be considered resistance until we don't change the dosage of rifampicin. So with the courage dosage, the majority of the RPOB mutations needs to be considered as potentially conferring resistance to be on the safe side. But we can discuss that more. I do have other two questions. One is from Dodge Lim from Philippines. What are the added value of genotypic DST at the programmatic level? Andrea Khabib, what's the current clinical utility of sequencing for informing the use of newer drugs? Dodge, I can call Andrea, is also next door, is not here with me, but I think Stefan can give you the answer.
I mean, so in our clinic now we are currently having the prospective trial where we use sequencing of, I mean, sputum-based sequencing for diagnostics of MDR patients, and they basically get the medication based on the sequencing result. That's basically that. I mean, it's like what can I say? I mean, that's of course then the full diagnostics is based on sequencing, and once we have finished our pilot trial, we most likely will switch our whole the whole testing in our reference laboratory for at least first-line drugs and second-line drugs to a targeted sequencing assay. And the algorithm for that may, for example, be that if you do not find with a targeted sequencing assay any resistance in first-line drugs, we would classify the case as susceptible, treat as susceptible, and go on. If we have MDR detected, we would look into classify resistance and susceptibility based on the result, and then go forward with, of course, with a short culture and a whole genome sequencing for the full sample. So I think it's clear that algorithm will work, and there may be very few strains at the end where we will still need phenotypic assays, maybe mix and other assays, but such an algorithm is of course desirable for new drugs.
I would like.
Yeah, so just for new drugs, for beta-caline, we now have or developing a pretty good catalog in Cryptic that should allow to classify the majority of patients with RV0678 mutations, and I think it's the only way to go. For Delamanid, I think it's a similar situation that I mean the only molecular assay will be sequencing. And for all new drugs now, we basically I think from the beginning of the development of the drug and when the drugs go into implementation, we should have, of course, a phenotypic assay, but we should directly also move into a wide testing of generated mutants that allow us good estimation of the drug resistance mutations.
I fully agree. Leen, do you want to add something?
Yeah, I just want to add, I would encourage everybody to do both phenotypic and genotypic testing for these new drugs, as it is, we are still having insufficient data on that, and we can only build data by testing. So I would encourage to do both new and repurpose.
Yeah, me too. And in addition, with rapid testing, we were able to really detect strains that were potentially resistant to bedaquiline when they have relevant indel in RV678. Those strains end up with an increased MIC, and some of them really fails therapy. We do have a question from Margaret. Hi, Margaret. Nice to see you here. She says, great presentations. Any comments on pyrazinamide phenotypic DST resistance, whole genome sequencing, no resistance mutation detected, and the EAI lineage?
Yeah, there are some other genes you can look at, of course. So you can also drop me an email, so we can look into that. Otherwise, I think Leen can maybe comment on that. I think especially pyrazinamide resistance is also error prone with false resistance result, right?
Yeah, especially in the beginning of the era of midget PZA testing, because that's the only medium that is recommended phenotypic testing. There was quite some false resistance and a little bit of false susceptibility, but this was mainly due because of less careful inoculation of the bacterial suspension. And if you really do it in a meticulous way, then you can overcome that. But it asks a lot of training and so on.
So
and that
also
the last
for low frequency. So I forgot that low
Also
frequency.
referring to EAI lineage. So we had few reports, and I remember we have done some study. They were also reported in the UK. In fact, there are some PZA resistance in specific strains of this lineage that are not associated with any mutation. We have done the study with Grace Smith. And I don't know, Margaret, at which point the study stopped, I think with the COVID, and we need to look back at the data. Many things stopped, and we do have another
question.
May
I just add, sorry, may I add, if you do midget PZA, you have the full resistant ones, clear resistant ones, but you also have the ones that are called resistant or susceptible, but they have some growth units, and we systematically always do sequencing to confirm the resistance in case of these. And that's why in the...
If I remember correctly, we did the MIC on those, and the Grace also sent those here for MIC. And yeah. I can get back to you on that, Margaret. We do have another question from Uladzmir Antonienka. Could we use cefazolin resistance as a surrogate marker for bedaquiline resistance?
I'm out. No, I don't think so. So clofazimine resistance, if it's...
clofazimine, no.
Yeah, if it's
not
the RVU678?
Not completely.
Not completely.
Yeah, it could be, we do have some cross resistance on 678, but not completely. So still phenotypic testing is seen. When we see mutation appearing, the MIC for clofazimine also goes higher. Not always goes to the level of resistance, though. But this is still an open question, I would say, at least for me.
So it's a partial cross-resistance.
Yeah, so it depends on the resistance markers, of course, but what we see in the clinic is if patients are pre-treated or with clofazimine, that are the ones that also develop resistance to bedaquiline later on, because clofazimine selects actually for RV0678 mutation, and that is cross resistance. But if you have freshly developed resistance under beta-killing treatment, then it can also be other mutations. And this are then the classical ATPE resistance mutations that lead to resistance to beta-killing, but not to clofazimine.
Stefan, the last question. This is from Arash. I'd like to ask you comments regarding to some important challenges of application of whole genome sequencing, such as the lack of standardization of whole genome sequencing analysis, pipeline, and genomic distances for defining clusters.
Okay.
You have 3 minutes to answer because at 3:15 we need to close.
So you may have seen some of our last publications on that. So together with Dick and Daniela, and we are aware of that. And we actually, it is essential if you build up your pipeline, that you validate your pipeline with a reference data set that is provided by another lab, for example, where the data, for example, the SNPs you should call in comparison to H37RV is defined. So I would always do that, and or you download a predefined pipeline. So I can only say download MTBSEC that has been validated across, for example, the Public Health England pipeline, and was also validated in the publications I just mentioned. And if you then implement it correctly, it should also result in a very conservative data analysis. And there is a potential risk when you use the wrong tools, that you overestimate SNPs, that you just call randomly false positive SNPs. This is, for resistance prediction, a real big problem, but also for SNP distances for cluster determination. It's clear that the distance you allow for a cluster, like 5 SNPs or 10 SNPs, whatever, is depending on your pipeline criteria. And in our experience, that needs to be validated beforehand. It is possible, but I would actually take advantage from a pipeline that is existing, or if you do your own, look for a lab that is doing that and then compare your results. So I think that's crucial.
Thank you very much, Stefan. And now, if you can, we can end by thanking everybody for attending, the speakers, Becton Dickinson for supporting us with this work. And we will meet you next week for the seminar of Jacob van Ingen on non-tubercular mycobacteria. I think this is a very hot topic, so I encourage everybody to attend. Thank you very much to all, and have a very good afternoon. Thank you, and stay safe.
Bye-bye.
Bye.
Bye. Thank you.